Okay. I'm gonna make a YouTube of this chapter covering the beginning that we did last week. So the objectives for this chapter are simple. Label a figure of the major stages of the cell cycle. We went over the cell cycle, including interphase, and then mitosis, and then also meiosis. Compare and contrast the cell division by binary fission, by mitosis, and by meiosis. Distinguish sister chromatids and homologous chromosomes. We all have homologous chromosomes because we're diploid, one from mom, one from pop. Diagram using the appropriate terms, the steps in mitosis and meiosis. Explain the importance of the checkpoints in the cell cycle. Identifies ways in which meiosis and fertilization together produce genetically diverse offspring during sexual reproduction. Okay, so the cell cycle is a sequence of events that makes up the life of a typical eukaryotic cell. A eukaryotic cell, remember, has a nucleus and organelles like mitochondria, ribosomes, other organelles. Organelles are separate entities within a cell, usually in the cytoplasm. Whereas bacteria and archaea don't have organelles. They don't have a nucleus. They just have a cell with a cell wall and a cell membrane, and the DNA is inside it. But they do have ribosomes. But they don't go through the whole process of using a nucleus and then the cytoplasm. Cells divide for a number of reasons, two main reasons: to reproduce. So organisms need to reproduce, and if they're either archaea or bacteria, they reproduce by nuclear fission, where they make two copies using mitosis, or also for meiosis in sexually reproducing organisms. They use that kind of cell division for sexual reproduction. And then once there's a diploid offspring, in this case a zygote, let's say one sperm and an egg, that's a fertilized egg, then it undergoes mitosis, where all the cells, it copies cells over and over and over again, and then eventually you get cellular differentiation. And during the copies, all cells end up with the whole genome of the organism, even if they don't use all those genes. And then finally, there is a dark side to cell division. Cells, except for germinalized cells, basically give up their right to reproduce. In multicellular organisms, and if they didn't do that, all hell would break loose and cells would be reproducing all over the place and you'd have cancer everywhere. So sometimes that happens, and then you have cancer, but for the most part, your body has ways to recognize cancer and then stop it. Okay. So we have a fertilized egg on the left. I think we have an 8-cell cell mass after fertilization. And then we have a baby following birth. and getting from the egg on the left, the fertilized egg, you undergo billions of rounds of mitosis and cellular reproduction. Okay. We talked about bacteria and archaea earlier. As you remembered, okay, there are 3 domains. It used to be thought that there were bacteria and then there were eukarya. But they discovered archaea, and archaea, and archaea and bacteria are called prokaryotes. But they actually, in terms of evolution, are not more closely related to each other than archaea and eukarya, because they share a more recent common ancestor compared to this older common ancestor right here. Okay, so we use prokaryotes to describe living cells without a nucleus. And at this point, we recognize 2 distinct groups, archaea and bacteria, that are like that. But there's another group that does have organelles, like a nucleus and mitochondria and chloroplasts in some, and that happens to be eukarya. But it turns out, in terms of evolutionary relationships, that group shared a more recent common ancestor with archaea more recently than with bacteria. But eukarya actually has bacterial genomes in it, in the form of mitochondria, which are actually bacteria that were engulfed billions of years ago. Okay. So, on the left is bacteria. It's got a cell wall, a cell membrane, so it's a cellular life form. It's got flagella. It's a kind of bacteria. It's got ribosomes. It's got DNA in the cell, but not in the nucleus, in the cytoplasm itself. Okay? And then, on the right, we have eukarya, which is the kind of cell that we're made of, that has a nucleus. It has organelles in the cytoplasm, like the mitochondria. It's got a whole bunch of other stuff. It's got ribosomes for turning RNA into proteins, but you can see it's got a lot of things. Okay. Okay. You can also have reproduction using what's called binary fission. And this is generally what bacteria and archaea use, where they copy their DNA. So it occurs in prokaryotes. Remember, though, prokaryotes are bacteria and archaea, but they are not more closely related to each other than archaea is to eukaryote. Okay, but this is how prokaryotes, bacteria and archaea, actually reproduce. They undergo mitosis. They copy their DNA, they copy everything else in the cell, and then the cell undergoes division, and there are two sister cells produced, and there are two identical daughter cells unless there's been a mutation during cell replication. But in general, they're identical clones. That means they have the exact same DNA, including alleles, in other words, the same versions of genes as their parent did. Why is binary fission referred to as asexual reproduction? Well, there's no sex. No gametes are produced. There's only mitosis, not meiosis. And so you don't get the diversity generated that you do with meiosis. We go back. We notice that bacterial and archaea both have circular chromosomes. But, eukaryotic cell division is a lot more complex, obviously, because remember the eukaryotic cell, it's got a nucleus, it's got ribosomes, it's got mitochondria, some have chloroplasts, it has all these other stuff like rough ER, Golgi apparatus, all this kind of stuff that has to be duplicated. Furthermore, eukaryotic cells have linear chromosomes. They're not circular. In cell replication, their cells have a life cycle in eukaryotes. For the most part, they stay in one part of that life cycle for most of their lives, doing what that cell does. If it's a muscle cell, it makes the proteins it needs for that cell, and it does all the muscle functions. So muscle cells are contractile, they operate in a certain way. The same thing happens with lung cells or blood cells or whatever. Okay, so most of the time there's this major stage called interphase that a cell resides in, where it's just doing its specific function depending on what kind of cell it is. And in fact, it's a specific stage in interphase in which that hangs out. And then the last phase of cell division in mitosis is the last stage in the cell's life cycle, where the DNA has to be duplicated and divided into daughter cells, and one cell becomes two, as I've kind of shown in the past, but we'll get into it more specifically. Okay, so this is the diagram of the cell cycle. During G1, most of the cell's life is basically in G1 or G0. We'll get to that in a second. This is where the cell does what it does. It Either if it's a muscle cell, like I said, it functions as a muscle cell, makes all the proteins it needs, all that kind of stuff. During interphase, it's called gap phase. It's the first gap phase. You don't have to know why. Now, when a cell is going to divide, it needs to actually copy all its DNA, not just the part that cell needs, like a muscle cell. It copies the whole genome, and that's called S phase. Okay, right there. And then finally, during gap phase 2, the cell grows, and it also copies all the mitochondria, all the ribosomes, all the machinery that it needs for the second cell. And it has to have enough energy to do that, and so it grows until that certain point, and once all those conditions are met, it goes into mitosis, which is a copying of the genetic material, the DNA. And this is called mitosis. There's 4 phases: prophase, metaphase, anaphase, telophase, and then finally you end up with 2 cells during cytokinesis, where the cells differentiate into separate daughter cells that are identical. Okay, now here's our diagram again. Most of our cells spend most of their time in the G1 phase, but sometimes cells need to divide. Like if they are worn out, they need to be replaced, or they need to be repaired, whatever, certain kind of tissue. They receive a signal that it's time to replicate. Then it goes into S phase, then it goes into G2 phase, and then mitosis and cytokinesis. Okay? But it needs a signal to do that. A cell can stay in G1 for a long time, or GO. Some cells types receive signals, start dividing when needed. So cells have different lifespans. Like skin cells, the outer layer of skin cells are replaced every few days, for that matter. Other cells are hardly replaced at all, like neurons. Okay. Now, once it gets in G1, if it gets a signal, it goes into S phase, in interphase. And interphase is G1, S phase, G2. The S phase, the chromosomes are copied and condensed in preparation for cell division. So, during gap phase, they're uncondensed, as you can see on the left. And once they're synthesized, you end up with 2 identical strands. Each chromosome is replicated before mitosis actually begins. This is S phase. And then at the beginning of mitosis, we actually see complete condensation of the homologous chromosomes, and that is necessary for mitosis to occur. DNA is usually either uncondensed or only partly condensed in the cell, in the nucleus, because all the genes, if it's all locked up, in tight packaging. The genes cannot make copies of themselves into RNA and then transfer that out into the cell cytoplasm and make proteins. Okay, so they start to partly condense during S phase. They fully condense for mitosis. Okay, so here we have the condensed DNA, and the whole chromosome is the whole bit. This whole thing, actually. Okay. The chromatids are these separate things, one from mom and one from pop. And then the centromere is in the center that ties them together. So sister chromatids are one of the condensed chromosomes. One from mom, one from pop. Okay. And here's mitosis. Here's the start of it. This is what the chromosomes actually look like. Here, you can see again. Okay. Now, as I mentioned in interphase, there's actually something called gap phase 0, and this is a non-replication phase, and there's some adult cells that never undergo replication. And so they stay in what's called GO phase. This phase can last for a few days to the rest of the cell's life. In other words, all they do is their function. They do not replicate and undergo mitosis. So, when is DNA replicated during the cell cycle? We talked about this, S phase. When is the cell cycle, does DNA separate into two genetically identical daughter cells? That's called cytokinesis. Let's, okay, and then if a cell, when the cell cycle separate, the DNA separates and you have two daughter cells as mitosis and cytokinesis. If a cell is not destined to separate in the daughter cells, what phase does it enter? GO. Okay. Okay. So, animation. Please view the cell cycle animation in Canvas for Chapter 6. It says cell cycle. Okay. Now, remember in cell division, prokaryote cell division is different. Well, it is different than eukaryotic cell division. But in both cases, you undergo mitosis and you end up with daughter cells. But in prokaryotes, namely bacteria and archaea, you end up with clones or identical daughter cells. So it's single-stranded, it's double-stranded DNA, but it's circular DNA, okay? And then the cell replicates, the DNA replicates, and other things replicate. The cell starts to divide, and then each daughter cell has an identical complement of the full genome of let's say, the bacteria in this case. Name one similarity between cell division in prokaryotes and cell division, eukaryotes, mitosis. Name one difference between cell division in prokaryotes and cell division in eukaryotes. Let's see. Well, prokaryotes don't have a nucleus, so they don't go through all that rigmarole of separating the DNA. They do separate the DNA, but not in the nucleus. And for reproduction, mitosis is reproduction for prokaryotes, but from eukaryotes, it's sexual reproduction. Two types of eukaryotic cell division, mitosis and cytokinesis. That's asexual, produces identical daughter cells. Meiotic division consists of meiosis 1 and 2, and cytokinesis 1 and 2. This is sexual reproduction. You end up with haploid gamete cells that are unique. In this case, either sperm or egg, depending on whether it is a male or female organism. And then one fertilizes the other, and you come back to diploid organism. Instead of during meiosis, you go from diploid at the beginning to haploid, and then fertilization haploid to diploid. Mitosis consists of 4 main phases. Inaphase, 3 parts, G1, S, G2, and then mitosis, prophase, where the nuclear envelope disintegrates at the chromosomes before that condense. They line up along the middle of the nucleus, or where the nucleus was in the cell. It's called metaphase. And then they, in anaphase, the spindle fibers attach and pull them apart so that each new cell will have a full complement of the DNA. And then telophase, that's complete, the nuclear envelope reforms. And then cytokinesis, the cell membrane reforms, and you have 2 daughter cells. Cytokinesis, where you have 2 cells. The DNA decondenses, and the cell goes back into interphase. Okay, now, is mitosis for reproduction or not? Mitosis. If you're wrong, I don't care. How many say yes? What?
It is asexual.
It isn't, right? That's what you said?
It is.
It is, okay. Okay. These 2 daughter cells that are identical, is that right? Okay. In reproduction, when you get gametes, are gametes identical or not? Sperm and egg, no. Okay, so now let's think about mitosis. Is mitosis for reproduction? If you have 2 identical daughter cells, are they different or are they identical? If gametes are unique, if gametes are unique, gametes for sperm and egg, that's reproduction, then is mitosis for reproduction? Huh? Let's try it again. It's okay. This is exactly what everybody's wondering in this whole class. Mitosis takes 1 cell, copies everything, divides, and gives you 2 identical daughter cells. Identical. Okay? They have the same DNA, they have the same alleles, which is versions of genes, they have the same everything. So if a muscle cell undergoes mitosis, you end up with a muscle cell. Is that reproduction? No. Okay? Reproduction, sexual reproduction, only involves gametes. So mitosis does not involve reproduction. Meiosis involves reproduction. And we're going to get to meiosis now. Okay, I want to make that clear. And thank you for sharing with the class. See, I love it when people are misinformed, because you just learned something. We all did. And everybody in this class was wondering the same exact thing. Maybe a few not, but for the most part. Okay? So I actually like it. I myself like being wrong, because it means I'm going to learn something. Okay. So, now, okay, we'll go on. So, here we see, let's see what we can do. Okay, so we've got interphase, where these are actually kidney cells, unpacked chromosomes. Then in prophase, condensation of the chromosomes. All the chromosomes then line up during metaphase. During anaphase, they start to separate because the spindle fibers pull them apart. And then during telophase, they separate, and eventually the cell wall, the nucleus reforms, the DNA decondenses, and finally cytokinesis. Okay, listen up. I'm sick. Yeah. Someone in either this class or the other class came in and sneezing, and they gave me the flu. So I'm pissed off. Okay. So we're just going to go through this, and then I'm going to drop dead. Okay? Okay. I told you I'd give you a study guide, but guess what? No. What? I was laying flat on my back all weekend, okay? Okay. I know I'm blaming you, and it's really no one's fault, but I just feel like it. Okay? That's all. So, just take it. I'm sure you're going to get back at me anyway. So, okay. So, we basically went through mitosis. So, when does mitosis happen in terms of the cell cycle? Anybody? Stop talking. What? Exactly. After interphase. Okay. And what are the 4 parts of mitosis? They're actually up there.
Yeah, so
Okay, let's see if we can. Okay. So, first of all, there's prophase. Then, that's when the nuclear membrane disintegrates and the chromosomes condense. Then there's metaphase along the middle, right here. Then there's anaphase, right? Anaphase. And then there's telophase, where the cell membrane, the nucleus reforms. Each cell has exactly the same contents. They're homologous chromosomes. And then finally, the last thing is okay. Do all the cells in the organism enter each stage of mitosis at the same time? No. That'd be crazy. Just when certain cells need to be replaced or repaired, or during development. what happens between the end of interphase and early prophase that changes the appearance of chromosomes? They condense. What does the mitotic spindle do in mitosis? It attaches to the chromatids and pulls them apart. And attaches at the centromere. Okay. Animation 3 is mitosis. Please view that. Okay. First of all, okay, so we have cell division, and one of the things that cells do when they divide is differentiate. So let's say you have a zygote. Anybody know what a zygote is?
That's when the sperm and the egg are together.
Fertilized egg, exactly. So a zygote is diploid. That means it's got chromosomes from mom and dad, right? A sperm only has dad's chromosomes. An egg only has mom's chromosomes. Okay. But when you have the zygote, then how do you get a human being out of it? What kind of cell division are we talking about?
Meiosis.
Huh? Mitosis, right? So mitosis is cells that are identical to each other making copies of themselves. So the fertilized eggs, the zygote makes a copy. Now there's 2 cells. Those 2 make copies. There are 4 cells. Now there's 8 cells, 16 cells, 32, 64. Okay? Identical copies. But after about 64, that's the blastocyst stage, when the cell mass is embedded in the uterus, then we start getting differentiation, because as you know, we have all kinds of cells in our body, right? We've got skin cells, we've got blood cells, we've got white blood cells, part of the immune system. We've got neurons, we've got pancreatic cells, we've got lung cells, we've got muscle cells, heart cells, all kinds of different cells. All of those cells have to differentiate into the constituent parts so that we can have a whole body. So all of that is mitosis, completely, just mitosis. Now, the cells then get instructions in terms of differentiating, where, remember, every cell has a complete genome in the nucleus. All the instructions in terms of different kinds of cells that are made. But only some of those are expressed. So as cells differentiate, some genes get turned on in certain cells and not others. And then cells differentiate into muscle cells, into bone cells, into all the different kinds of cells. That's all mitosis. Okay. Now, okay. Cell division in cancer. Okay. Now, did we go over checkpoints? Cell checkpoints, the cell cycle? Well, we'll go over them quickly. So, as you might remember, there's the cell cycle, there's interphase. We have these checkpoints, and the cells have to pass through them. But sometimes you get mutations. And cells decide to go off on their own and start replicating like crazy. Remember, all your cells in the body, they've basically given up reproduction on their own, right? All for one, one for all, so to speak. The only cells that are involved in reproduction are the germline cells that give rise to the gametes in meiosis, which we're going to get to now. But in the meantime, cancer is one of those situations during mitosis. As cells divide, sometimes there are mistakes made. One mutation isn't usually going to cause a cell to become cancerous. It's usually a series of mistakes. Now, remember, there's usually checkpoints along the way when they go through this division process to stop them if they're not right. And for the most part, that's why we're not riddled with cancer. Okay. Hold on. Some hormones and growth factors act like the gas pedal on a car that make cells divide. They get the right signal. They get enough energy. Others, hormones act like brakes. They stop cell division or prevent cell division. Okay, so here we have a cell. We've got G1. A lot of cells go into GO where they basically never change. They don't go through mitosis after that. But some cells do. They're in G1, and they do most of their functions, and then they get a signal that tells them, oh, it's time to undergo cell division, mitosis. It might be because of repair, cells wear out, or damage, or it might be because of development, you know, with the zygote and all that. Okay, so the cell cycle, the interphase part in the green, the cell has to undergo those three phases. G1, S for synthesis of the DNA, and then G2. But there's checkpoints along the way. It's kind of like, we don't live in a country like that, but in some places there's military checkpoints, and you have to go through, and if you have the right papers, they let you pass. If you don't have the right papers, they shoot you. It's as simple as that. Wonderful system. Same thing with cells. If everything is all good in terms of their papers or in terms of their growth rates, there's a whole bunch of different signals they give. They're allowed to pass through into S phase, which is synthesis of the DNA. That means all the DNA is copied. The whole kit and caboodle, because remember, all cells have the full complement of DNA for the whole genome, not just the genome, the part of the genome they need. Then they go into G2. In G2, they're getting ready to divide, so they need twice as much of everything, including mitochondria, ribosomes. They need another nucleus, everything, and lots of energy to do all this and grow. So when they get to end of G2, there's another checkpoint. And it undergoes the same sort of thing. They go through, and if everything's good, they go on into mitosis, where the nucleic acid in the nucleus and the DNA is all copied. And so both cells have identical DNA, okay? And then there's also a metaphase checkpoint in mitosis. Okay. Now, there's something called a tumor suppressor gene. So, as you can imagine, cancer in a multicellular organism might become a real problem. And it is. In fact, cells there's mutations happening during mitosis all the time, and that has to be stopped. And so there's this whole elaborate biochemical system for recognizing cells that are not acting properly and then stopping them. And this is called tumor suppressor genes. There's a whole bunch of different ones. You might have heard of one of them. There's the BRCA tumor suppressor gene. If there's a mutation in that, women are much more likely to get breast cancer or uterine cancer, and men are much more likely to get prostate cancer. So this is a system where the tumor suppressor is supposed to stop cells from dividing and to actually commit suicide because those cells are not behaving properly. But if it's broken, if there's a mutation in it, it doesn't do their job. Okay. Back. Okay, you can answer these on your own. Now, in terms of cancer, this is all we're going to cover. You end up, let's say you end up with cells that are reproducing with no control. They're going through all the checkpoints, everything. Maybe because there's a tumor suppressor mutation that's not stopping them, whatever. And you end up with a cell mass by rapid runaway cell division, mitosis. That doesn't mean you have cancer then. That means you have a cell mass and it might be benign. And benign means that it might keep dividing slowly, but it stays in place. It doesn't invade nearby tissue. It's only when cells start invading nearby tissue that they actually become malignant or cancerous. Okay? So, and that's called losing contact inhibition. So when cells are next to each other, that usually sends a signal, don't divide. Okay? It's called contact inhibition. Some cells lose that in terms of a mutation, and they continue to divide and grow into those adjacent cells. Another thing that you probably heard about is metastatic cancer, where cancer cells travel all over the body. That's another kind of mutation that happens in some cancers, where they normally they have this adhesive function that makes them stay in one place. Once they settle in a certain place, let's say a muscle cell or a pancreatic cell, they don't go moving around. But occasionally they lose anchorage independent, or they lose anchorage independence. In other words, they suddenly can just go off and gallop and decide it's time for a nice little jaunt around the individual. Okay. When cells break away, they invade other tissues from loss of contact inhibition, and they form different cancers all over the body in different areas, but from the same cell type as the original tumor. And then another thing that cells do is cells need a blood supply, and so cancer releases growth factors so that the body starts developing blood vessels that can supply the tumor. Okay. Okay. So that's enough about cancer. It's a bummer. Don't get it. It's better now. Earlier detection. But there's still some cancers that are really, really bad. But it's a molecular process, and hopefully, and a genetic process, and hopefully in the future, they'll get better and better at curing it, and they already are. Meiotic division. So meiosis is sexual reproduction. That means, unlike meiosis where you have identical daughter cells, in meiosis you have unique gametes. and we're going to show how that actually happens. And the gametes, of course, that we're familiar with are sperm and eggs. Plants have pollen and ovaries, okay? But same sort of thing. Gametes, so we're diploid. We have a full complement of DNA, one identical chromosome from our father, one from our mother. These are called homologous pairs of chromosomes. But our sex cells, our gametes, only have a single chromosome, and so they're called haploid. Okay. Okay, so vocabulary. Our somatic cells, that means our general body cells, they never give rise to gametes. Okay? They're diploid. They have 2 of each chromosome. Each pair is homologous. They have the same genes in the same places on the chromosomes. It doesn't mean that each gene is identical. They might have different alleles. So there's different versions of a gene. There's we'll say this, there's a locus, hair color. We'll call it that. But you might have an allele that makes it more likely that you have a pigment for brown hair or for black hair. Or you might have one for blonde hair. Or you might have a mixture. So alleles are different versions of genes. Okay. Meiosis produces gametes. That means there's germline cells that I talked about, testes and ovaries. And during sex, one gamete meets the other. They fall in love. They produce a baby or a zygote. A baby's way down the line. And by the way, the egg is, 10,000 times bigger than the sperm. Sperm have nothing. They're just little things with DNA with a tail that they can swim along and go places finding an egg. An egg has mitochondria, ribosomes, all those other stuff that's in the cytoplasm. Okay. And then when you have the gametes coming together for fertilization, you restore diploidy. In other words, now you have a zygote that has male and female, or mom and dad's chromosomes. Okay. So is the zygote haploid or diploid? Zygote, remember, sperm and egg. Diploid. Haploid, each sperm is haploid and each egg is haploid. When they come together and they're fertilized, diploid. Good. Like I said, I love it when people are wrong because most people get it confused. And if we don't bring that up, then they go on not understanding. Okay. So, which cellular process creates a baby from the zygote? Meiosis or mitosis? Mitosis. Okay? We've already done meiosis. We've got a zygote. The zygote is now undergoing cell division. That's mitosis. If a mother or father, forget about that one. Okay. Okay. So, meiosis produces daughter cells, just like mitosis. They're certainly related to each other. And there's 2 stages of meiosis, meiosis 1 and meiosis 2. And we'll go through them, and we'll see a little video on that. Now, meiosis 1, the cell has, remember, in interphase, the germline cell has undergone S, where they synthesize all the DNA, so they have twice as much DNA. The division, just like mitosis, separates homologous chromosomes, And those chromosomes, though, have twice as much DNA. The cells, though, are haploid, not diploid. Even though they have twice as much DNA, they have sister chromatids, and those are going to further split off into either a sperm or an egg. And then they remain together. So this is all talk. We'll take a look at it in a second. But just so you know, meiosis 2, we've already got twice as much DNA. But in meiosis 2, it has half the chromosomes, each cell does. And this division separates sister chromatids in the cytoplasm into 4 different daughter cells. So let's go back. Meiosis with cytokinesis creates haploid daughter cells. What is a haploid cell? A haploid cell has only one chromosome. Not two. We have a diploid. So during meiosis 1, we start out with a haploid cell on the left. Okay. The chromosomes line up along the metaphase plate. They start to pull apart, and you finally end up with 2 haploid daughter cells, but they're homologous chromosomes with chromatids. Okay? And then, finally, during meiosis 2, same thing, but you end up with single single chromosome gametes that are unique because of crossing over and independent assortment. Sorry. Okay. So let's go back. Is the daughter cell haploid or diploid? After the first meiotic division, is haploid. After the second meiotic division, is still haploid. So what's the difference between homologous chromosomes? Homologous, homo is same. In this case, the chromosomes are identical. They have the same genes in the same places along the chromosomes. The chromatids are the independent chromosomes that are identical to the one next to it, part of the homologs. Okay. let's see. We could go back. I think we'll go back. If you go back to okay, here we go. Now we're on to the next. Meiosis with cytokinesis creates haploid daughter cells again. Question 3. The skin cells of a house cat contain 38 homologous pairs of chromosomes. How many chromosomes are present in the egg cells they produce? Okay. How many chromosomes are present in the egg cells they produce? And that's 38. They are not homologous. They are 38 chromosomes individually. Instead of, in a diploid cell, they are homologous pairs. So here's back to these slides about what a homologous chromosome is and what diploid is. On the left, we have in A, homologous pairs of chromosomes. They each have the same genes, but different alleles. Like the one on the left has big A, the one on the right has little a, and then big B and big B. They're identical for both on the left and right. And then again on C, there's two alleles. When you have a diploid organism, there's always going to be two alleles. Sometimes they're identical, they're homozygous. Sometimes they're different, heterozygous. We'll get to that later. Okay, so the homologous pair of chromosomes is this, right? This is the pair of chromosomes. And it asks if a cat has 38 homologous pairs of chromosomes in its skin cells. So we have 23. Here we are over on this side. See, cats have 38. And remember, when you're undergoing meiosis, you end up with half the number of chromosomes total, but the same number of actual chromosomes. So if you have 38 homologous pairs, each gamete has one pair, and so there's 38 chromosomes. Actually, okay. So humans, we have 23 pairs of chromosomes, 22 are identical for the somatic cells, and then we have two sex chromosomes, an X and a Y. A female is XX. They get one X from their mom, one X from their dad. Or a male is XY. They get one X from their mom and a Y from their dad. Okay. Now, when eggs and sperm are made during meiosis in the germline cells, the testes of the ovaries, they only have one chromosome instead of two. They have chromosomes from either their father, their father's father, or their father's mother. Okay, so we'll get to that. And they're haploid. Eggs and sperm have one of each chromosome. These cells are haploid, either in a sperm or an egg. Okay. Here you can watch the animation for meiosis, please, on your own time. They go kind of fast, but they go through everything. Okay, here we go. Genetic diversity. Okay, So we're talking about sexual reproduction, and we're talking about sexual reproduction in terms of genetic diversity. If an organism lives in a very stable environment that never changes, it should become very well adapted over time. And then any further genetic change should not happen because it's already as well adapted as it possibly can be. But we don't live in that kind of environment. When an environment is the same all the time, we say it's homogeneous. But an environment, most environments are heterogeneous. Spatially or temporally. Now, those are big words, and so heterogeneous means different. So an environment that is different spatially means in terms of space or over space. So you might have one kind of habitat in one area, and then you might have next to it an adjacent habitat that's different, or further away a different habitat. That's spatial heterogeneity. And temporal is time. So obviously during seasonal changes, things are different between the summer and the winter. Now, since the environment is heterogeneous in terms of differences in habitat and different time periods, some alleles, versions of genes, are going to be good in one habitat and not the other. So if an organism lives in a diverse environment or a changing environment, it's good to have lots of genetic diversity. Okay, so that's one reason why we actually have sexual reproduction, because if you remember, during bacteria reproduce by binary fission, where they make identical copies of themselves. No genetic diversity, unless there's actually a mutation. Whereas sexual reproduction mixes things up because you've got two different genomes in an individual who's diploid. You've got the mother's genome and the father's genome. And that diversity might produce an offspring that, let's say, gives it an advantage in certain habitats and times versus others. And that kind of diversity in response might be beneficial. So, meiosis occurs in sexual reproducing organisms. Half of an organism's DNA comes from a different parent, generally unrelated. Meiosis and fertilization maintain the constant chromosome number of a species, but create genetic diversity within a population because of all this mixing up from a number of different things, including crossing over, independent assortment, and random fertilization. Okay. Sexual reproduction. Crossing over and independent assortment happen during meiosis 1. Okay? First of all, we can look at crossing over. We see the blue and the red exchanging so that this chromosome from, let's say, the father comes from his father's father with a little bit of his mother thrown in because of crossing over. And the same thing, this maternal chromosome that this fellow had got a little bit of paternal chromosome in there because of crossing over. Or it could have been the female, an egg, in which case, same sort of thing. She's got a father and a mother. Okay, now, so crossing over happens during metaphase 1, and then, as you can see, the chromosomes can line up differently. So, here we have a red chromosome on top and a blue chromosome on the bottom. Or you could have a blue and red chromosome on the top, mainly blue, or mainly red on the bottom. Or you could also have, on the left, blue on top, red on the bottom. This is called independent assortment. Okay? So with crossing over, not only is there an exchange in the chromosomes of genetic information from that individual's mother and father, but then there's, depending on which chromosome goes to the gamete, it'll be a primarily maternal or paternal chromosome, but not all the chromosomes are individual. So it can be paternal for one chromosome, maternal in another, and then all that crossing over. And that happens in metaphase 1. Okay. And then if you follow it in metaphase 2, you see which way things go and which way they can go. And you end up with very diverse gametes. Here you have a gamete on top with mainly maternal chromosomes. Here you also have a gamete on the very bottom. So here's the top, here's the bottom, with mainly paternal chromosomes. And then here in the middle, you have a maternal chromosome, 2 maternal chromosomes, but with some paternal chromosomes mixed in, or mostly paternal chromosomes with maternal chromosomes mixed in. Very diverse. So you have 4 different kinds of gametes that can happen from that. Okay. So crossing over the physical exchange of chromosome segments between non-sister chromatids of a homologous chromosome occurs during meiosis 1, and it's an independent assortment. Okay. Crossing over produces chromosomes with new combinations. We have a maternal chromosome on top, up here on the left, and a paternal on the right. And when they line up along the metaphase plate, one arm of the paternal and the maternal crossover, and they switch or flip material so that the new chromosome, now, if it was maternal, now it has some paternal genetic alleles, or vice versa, if it was paternal, now has some maternal. Okay. And so here we are, and this is meiosis 1, metaphase 1 is when it actually happens, and we end up with chromosomes that, like this one over here, is primarily maternal. In other words, from the individual's mother, but also with a little bit of paternal mixed in. And this chromosome is primarily from the individual's father with a little maternal mixed in. And then you can see how these gametes end up. Why is a term crossing over appropriate for the exchange of DNA segments between homologous chromosomes? It happens in metaphase, where the arms of the homologous chromosomes, in other words, the chromatids, cross each other and then exchange genetic information. At what stage of meiosis 1 or 2 does crossing over occur? Meiosis 1. At what stage, what would be the effect of crossing over between two sister chromatids? They would become more diverse. Okay, now we're on to independent assortment. Forget about crossing over. Okay, so we've got homologous chromosomes in the top here. We've got maternal chromosomes, and paternal chromosomes. And they can go either way. So in meiosis 1, you can have a homologous pair of maternal chromosomes, like here, or a homologous set of paternal chromosomes, like here. Okay? Or option B, you could have a maternal set on the left here we go, and a paternal set on the right, or a paternal set on the left and a maternal set on the right. Each individual chromosome is independent and independently sorted compared to all the others. So we've got 23 pairs of chromosomes. You could have maternal, paternal, maternal, paternal, or you could have maternal, 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 paternal, maternal, paternal, 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 maternal, right? Doesn't matter, it's random. And because of this random assortment, and this assortment happens in meiosis 1, okay,